Okay, we're gonna make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at the cell mediated response. Okay, so last lesson we looked at phagocytosis, which was the first response that your body makes in order to counteract the pathogens which have entered the body. In today's lesson, we're going to be looking at the second line of defense after the phagocytes have done their job and they are now sort of um, so the phagocytes have done their job. Now the body needs to make antibodies and sort of take the pathogen away, okay, and make you feel better. And that's what the cell mediated response will be. Now we're not going to we're only going to be looking at T cells today. So the T cells part of the response, and then in our next lesson we're going to finish off the response by looking at the B cells, and we'll have a look at exam questions in our next lesson of what they can ask. Okay, we're going to basically look at the first half of the process in today's lesson. Okay, so again, as always, here's the specification, and we're basically following the specification from top to bottom. Okay, and in specific, in today's lesson, we're going to be focusing on this part in terms of T lymphocytes and how they respond to antigen presenting cells and phagocytes, and what the different types of T cells there can be. So there can be T helper cells and cytotoxic T cells. Those are the key T cells that you need to be aware of. Okay, so if we make a start, so first thing first, over here we have a recap of an exam question that we looked at in our last lesson. So last lesson, we looked at this exam question towards the end of the lesson, and I showed you a model mock scheme answer for phagocytosis. Okay, and I'm just going to recap what happens in phagocytosis very quickly, so we know what stage we are on currently. So remember, a pathogen enters your body, and the pathogen releases certain chemicals, and it releases sort of certain sort of substances which attract phagocytes. Okay, and phagocytes are a macrophage, which is a type of white blood cell within our body. So the phagocytes, they are attracted to the pathogen because of the chemicals the pathogen has been releasing. Now, once it's attracted to the pathogen, there are receptors which are present on the phagocyte, and what will happen is that the antigens are present on the pathogen. It will move towards the phagocyte, and the phagocyte will engulf the pathogen. Okay, so the phagocyte engulfs the pathogen, and once the pathogen is within the body or within the phagocyte, it fuses with a lysosome. Okay, so lysosomes move towards the phagocyte. It is firstly contained within a phagosome vesicle. The lysosomes, they fuse with that phagosome vesicle, and then the lysosomes release hydrolytic enzymes known as lysozymes, and the lysozymes, they destroy the pathogen. Okay, they destroy the pathogen, and any useful substances that the pathogen releases will be used by the phagocyte itself. The antigens, which is just, remember, we looked at the definition in our last lesson as well. An antigen is a foreign protein which stimulates an immune response. The antigen will be presented on the phagocyte, and the phagocyte would become an antigen presenting cell. Okay, so that's the point in time which we got up to. And the purpose that I've put this exam question as a recap is just so you can get familiar with the mock scheme and the process of phagocytosis. So this is an application exam question. This is not a direct phagocytosis exam question, but as you can see, even for application exam questions, the mock scheme is exactly the same. Okay, so a cell ingests or engulfs the antibody. The lysosomes fuse with the phagosome vesicle, and then the lysozymes, which are released from the lysosomes, they break down the antibody and the ADC to release the drug. Okay, so this is the application version of the phagocytosis exam question, which we looked at in our last lesson. But now we've had a recap of the process of phagocytosis and the sort of processes, the steps that you need to be familiar with. We're now going to move on to the cell mediated response. Okay, now before we do that, you need to be familiar with this keyword. Okay, and this keyword is known as lymphocytes. Now, lymphocytes are basically white blood cells. They're a type of white blood cells which are also part of the immune response. So we saw that phagocytes, they are part of the immune response. Phagocytes are an example of a macrophage, and that macrophage is an example of a white blood cell. Okay, now lymphocytes, they are also an example of a white blood cell. Okay, and they will also trigger the second part of sort of antibody production and immunity. Okay, now first thing first, you need to know where lymphocytes are made. Now, all lymphocytes, they are made in the bone marrow. Okay, so in the human bone marrow, that's where all your white blood cells, your lymphocytes, they are made. Okay, now in today's lesson, we're going to focus on T cells. Okay, and T cells, they mature in the thymus. So different location, they are made in the bone marrow and they mature in the thymus. Okay, so that's different because you also need to know about B cells. Okay, and B cells, they don't mature in the thymus. B cells, they mature in the bone marrow as well. They are made in the bone marrow and they mature in the bone marrow. T cells, T for T cells, T for thymus. Okay, so just keep that in mind if you want a memorization strategy. So they are made in the bone marrow and they mature in the thymus. Okay, and the thymus is basically sort of the part that you have sort of between your lungs. That's where your thymus gland is. Okay, so that's the part where they mature. Okay, now a cell-mediated response, that's a response which involves T cells and normal body cells. There are also B cells that you need to be familiar with. However, B cells, they are part of a different response. They're still part of the process, but they're part of a different, the name of the response is a bit different. So this is a cell-mediated response. This is stage 2. So stage 1 is your phagocytosis. Phagocytosis is stage 1. Stage 2 is your cell-mediated response. And then stage 3 is your humoral response. And that's where your B cells come in. Okay, so as I said, over here, stage 1 is your phagocytosis. And over here, you have your phagocytes. Stage 2 is your cell-mediated response. And this includes your T cells. And then stage 3 is your humoral response. And this includes your B cells. Okay, now this humoral response we're going to be covering in our next lesson. Okay, but a cell-mediated response is one which involves T cells and normal body cells. Okay, now a cell-mediated response is specific. Okay, so it's very specific because T cells, they respond to the antigens on the surface of cells. Now let's break that down a little bit. Okay, now antigens are a foreign protein which stimulates an immune response. So a foreign protein which stimulate an immune response. That's what antigens are. And we know that after phagocytosis has occurred, the phagocytes, they take in the pathogen, they break it down, and the antigens which come from the pathogen, they will present the pathogen, they will present those antigens on their surface. So the phagocyte becomes an antigen presenting cell. So a phagocyte, it's taken in the pathogen. I have a good image now I'm going to show you. But it's taken in the pathogen. It's taken in the pathogen, it's broken it down, and it's used the antigens, and it's going to stick the antigens like that on the surface. Okay, just like that. And then these antigens, they are going to be activated by T cells. Okay, so this is the part where phagocytosis gets still. Then T cells, they will come and they will bind to these antigens that the phagocyte has taken in, or any body cell that has taken in these pathogens, any antigen presenting cell which it has found, it will go bind to that antigen presenting cell. Okay, that's why it's a specific response, because it only binds to the antigens that are present on those specific T cells. Okay, so those are your lymphocytes. Okay, now if we looked at antigen presenting cells in our last lesson, but if we just quickly just go over them again, an antigen presenting cell is any cell which can present a non-self antigen. Okay, and a non-self antigen is just an antigen which doesn't belong in the cell. So for example, when a pathogen enters the body, the antigens which are present on a pathogen, they are not known to the body. They are different antigens. So if a body cell, if a macrophage or a phagocyte presents the antigens from the pathogen, those are non-self antigens because the phagocyte doesn't recognize them. They don't belong in the body. They've come from a pathogen. So that's what an antigen presenting cell is. It's presenting antigens which don't belong in the body. Okay, and where do these antigens come from? Infected body cells, macrophages, cells of transplanted organs, and cancer cells. So that's why, if we think about it, organ donation, organ transplant can be risky. Okay, and it can be risky because the immune system can reject the organ. Why can it reject the organ? Because the reason why it can reject the organ is because the organ doesn't have the same antigens which the immune system from the person recognizes. If it doesn't have the same antigens, then the immune system is going to go attack the antigens from the organ. It's going to present it's going to break down the organ first, and once it's broken down the organ, the organ has gone through phagocytosis, is going to present those antigens, and then the T cells are going to combine to those antigens. That's why there's always a risk there with organ transplants. The immune system could reject the organ because the organ doesn't have the antigens that the immune system recognizes, and it will treat the organ as a pathogen which has entered the body. That's why people with organ transplants will have to take immunosuppressants for the rest of their life. Okay, because immunosuppressants, they will suppress the immune system from attacking the organ that has been transplanted. Okay, so that's a bit of application that you could get. And again, cell mediated T cells, it's because they only respond to antigens which are presented on cells. They don't respond to free floating antigens in the blood. Okay, so sometimes in the blood you can just have free floating antigens in your bloodstream. Because they're just floating in the blood, the T cells, they won't respond to those antigens. They will go specifically to the ones which are bound to a cell, okay, to a phagocyte or an antigen presenting cell. Okay, so that's a bit of an overview of what we are going to be looking at in today's lesson. I'm going to show you an image now about where we're at currently, and then we're going to go through the cell-mediated response. So in this case over here, this is the image I'm going to show you. So what we've had so far is that we've got a phagocyte over here. This big cell is a phagocyte, and the phagocyte has specific receptors, okay? And a pathogen has entered the body. The pathogen has specific antigens present on its surface. Those antigens, they are going to bind to the receptor of the phagocyte. So the pathogen is going to release some sort of chemicals that's going to attract the phagocyte, and then the phagocyte is going to bind through the receptors that it has to the antigens on the pathogen. And that's going to cause the phagocyte to engulf the pathogen. So as you can see, the phagocyte is engulfing the pathogen. Today's topic is the cell-mediated response. Okay, so engulfing the pathogen. Once it's engulfed, the pathogen is held in a phagosome vesicle. Okay, it's held in a phagosome vesicle, and these are lysosomes, and these lysosomes, they contain lysozymes. So what's going to happen, these lysosomes, they're going to go and they're going to fuse with this phagosome. Okay, and once they fuse with the phagosome, you don't need to know this name, but the lysozymes, these small dots, they're going to break down the pathogen. Okay, so they're going to break down the pathogen. So as you can see, the pathogen has been broken down, and all that's left is the antigens. Okay, so the waste products, these are your waste products. They're going to be released or used by the cell. And the antigens, they are presented on the surface of the cell. So the T cells and the lymphocytes, they can come and they can trigger the next part of the immune response, which is the cell-mediated response. Okay, so the phagocyte has become an antigen-presenting cell. Okay, so that's where we're at the moment. Now, the cell-mediated response. So once the pathogen has been engulfed and destroyed by the phagocyte, the antigens are positioned on the cell surface. This is known as an antigen-presenting cell. So that's the stage that we're at currently. The phagocyte has engulfed the pathogen, and the antigens have been are present on the surface of the phagocyte, which means that it's an antigen-presenting cell. Okay, now there are many different types of T cells. Okay, now the main one that is in the highest concentration within your body are helper T cells. Okay, and they're going to come and help this phagocyte that is presenting these antigens. Okay, so the second step is the helper T cells, they come round. Okay, they realize that this phagocyte needs help. Okay, so they're going to come, they're called helper T cells, and these helper T cells, they're going to have receptors on their surface. So the antigen presenting cell, the phagocyte, has antigens which are on its surface. These helper T cells, they're going to come round, and because these helper T cells have receptors on their surface, they're going to bind to the antigens that are present on the white blood cell. Okay, they're going to bind to the antigens present on the phagocyte, on any antigen presenting cell. So stage 2 is where your helper T cells come around. Now, what's going to happen is that these helper T cells, because they have receptors on them, they're going to come and they're going to bind to the antigen presenting cell. They're going to bind to the antigens that are on the antigen presenting cell. Okay? And that's going to activate the helper T cell. Okay, so they've bound to the antigen on the antigen presenting cell, and that's activated this helper T cell. And this helper T cell, because it's been activated, it's going to divide by mitosis. It's going to divide by mitosis, and it's going to create a large number of clones. Okay, so that's what's happening in your cell-mediated response. These helper T cells, they're going to grow in numbers. That's why if you get ill and you take a blood test when you're ill, you will realize that you have a high number of white blood cells. Okay, if you look at your blood test reports, you will see that in your body there is a high number of white blood cells. If you're suffering from cancer, one of the early detections of cancer is that you have a really, really high number of white blood cells. And that's because your white blood cells are trying to defend yourself against the cancer. Okay, so that's what you would see in a blood test. So that's why you see that spike in the number of white blood cells on your report, is because these helper T cells have been activated. They've been activated and they're dividing by mitosis so they can produce a large number of clones, and they can sort of get as much help as they can to defend your body against this pathogen. So that's what's happened. Okay, so the first part, we've got an antigen presenting cell. The helper T cells, because they have receptors on their surface, they're going to come and help. They're going to bind to the antigens on the antigen presenting cells. That's going to activate the helper T cells, and that's going to trigger them to divide by mitosis and produce a lot of clones of themselves. Okay. Now, once they've produced all of these clones, there are many different functions that these helper T cells can give the cells that have divided. It can give different roles to different cells. Okay, so these helper T cells have divided. And what can happen is that differentiation can occur. Okay, so we had one helper T cell which has been activated. This helper T cell will divide by mitosis to form many other T cells. Okay, and the T cells that are formed, they can differentiate into different types of cells. So some will remain as T helper cells, okay, because obviously you don't want to get rid of all the T helper cells that you have. And the reason why you have T helper cells still is so the B cells can be activated. Okay, the T cells, they activate B cells. Okay, we will look at that in our next lesson. But some of them will stay and they will activate the B cells so you can produce antibodies and sort of plasma cells. Okay, memory cells and antibodies. Some will stimulate macrophages to perform more phagocytosis. So there might not just be a little bit of pathogen, there might be a lot of pathogen within your body. So more phagocytosis may need to be apparent. So that means once these T helper cells have divided, some of them may differentiate into phagocytes to stimulate more phagocytosis to occur. Some of them will become memory cells for that specific shaped antigen, and some of them will become cytotoxic T cells. Okay, now cytotoxic T cells we're going to look at on the next slide. You need to know about cytotoxic T cells in specific. Okay, so that's cell-mediated response. Your helper T cells have been activated after binding to the antigen present on your antigen-presenting cell, and that stimulated the helper T cells to divide by mitosis and increase in number. These cloned helper T cells, they can now differentiate into different functions. So some will turn into phagocytes to carry out phagocytosis. Some will stay as T helper cells so that B cells can be activated. Some turn into cytotoxic T cells. Some become memory cells. That's what your T helper cells can become. Okay, and that's basically the end of cell-mediated responses. Okay, the next part of the response is the humoral response. Okay, and that involves B cells, and that's the final part of a response, final part of your immune response. Okay, that final part of your immune response we'll look at in our next lesson, along with some exam questions with everything, T cells and B cells. What we're going to look at now is cytotoxic T cells. Okay, so as you can see, some of the T helper cells, they become cytotoxic T cells. Now, cytotoxic are killer T cells. Their job is to kill the pathogen which has caused damage in your body. So there's going to be some pathogen just lurking around your body. It needs to get rid of all those pathogens and all the infected cells that are within your body. Okay, so cytotoxic T cells, their job is to destroy abnormal or infected cells. And the way that they do this is they release a protein called perforin. And perforin, it embeds into the cell membrane of the infected cell, and it makes a pore, it makes a hole in the cell membrane. And that means that any substance can enter or leave the cell. The cell has now been damaged and will be destroyed. Okay, so that's what cytotoxic T cell basically does. It destroys your abnormal and your infected cells by releasing this protein called perforin, so it can make a hole in the cell membrane to allow any contents to leak out, and so the cell would be destroyed. Okay, now putting this into an example, when you have a sore throat, when you have a cold, your cytotoxic T cells destroy the infected body cells in your throat. That's why when they're destroying those infected body cells in your throat, you will feel sort of, if you look at the back of your throat, it's a bit red. Okay, because your white blood cells are attacking and getting rid of all those pathogens which are present in your throat. And you will also feel like your voice is sort of croaking up a little bit. And that's because the cytotoxic T cells, they're damaging the infected body cells. You will initially feel when you're getting a cold, you will initially feel like, okay, I'm getting a cold. My throat is going to go, my voice is going to go, or something like that. You will have that sensation within your throat. And the sensation comes because your pathogen has entered the throat cells. Okay. Now, when the throat cells start getting damaged, and when the white blood cells, when the lymphocytes come in, and cytotoxic T cells, they come in and attack the throat cells, that's when you feel sort of your voice is gone. Okay, because your cells have gone because they've been killed by the cytotoxic T cells, and after a while, after your blood has replicated and your throat cells have come back, that's when your voice comes back. Okay, so that's an example that you can have in terms of sort of analyzing how it actually works when you get infected with a pathogen. Okay, so as I said, this diagram is the first part of how it becomes an antigen presenting cell. A phagocyte becomes an antigen presenting cell, and then your cell mediated response is when your T helper cells are activated because their receptor binds to the antigen which is present on this antigen presenting cell, such as this phagocyte. That stimulates the T cells to undergo mitosis and make a lot of copies of themselves. And when they've made this many copies of themselves, they can then tell different cells to differentiate into different things. So some will stay as T helper cells, some will differentiate into phagocytes, some will become into memory cells, and some will turn into cytotoxic T cells. Okay, and then cytotoxic T cells, they will destroy any abnormal cells or any infected cells by releasing the protein which was called perforin, and that perforin makes a hole in the cell membrane, which means that the cell has been destroyed. Anything can enter and leave the cell. Okay. So that's your cell-mediated response. That's the whole of your cell-mediated response. The final stage is your humoral response, which we'll look at in our next lesson, as mentioned. Now, exam questions we're looking at in our next lesson as well, but I've decided to put one common exam question over here. It says the destruction of T cells by HIV leads to the death of an infected person. Explain why. And the 2 marks that you are getting, we've actually touched upon this. But B cells are the ones which um, are the last stage of your response. B cells are the ones which create your antibodies. B cells are the ones which create your memory cells. Okay, now if B cells aren't activated, then B cells will basically, your immune system will not have sort of any form of defense against any other pathogens which enter your body. Okay, that's if B cells are not um, sort of made. Okay, if they are not able to make antibodies or memory cells, then that means any other pathogen which enters your body, it will slowly destroy all your white blood cells. Okay, such as HIV. HIV targets your T helper cells. Okay, your T helper cells means that you're not going to have enough B cells, you're not going to have any cytotoxic T cells, you're not going to have enough phagocytes. It's going to knock off all the immune cells that you have. And if your body isn't able to contain those immune cells, then it's not going to be able to fight off any future pathogens which enter your body. Okay, so that means that the person will be unable to fight off any infections which come in the future. Okay, which makes them more susceptible to passing away, to dying. Okay, that's why HIV is a very dangerous disease, and it needs to be cured very quickly before it gets to the latest stage of AIDS, where your body can no longer fight off any infection, any new infection which enters. Okay.